Dr. Crowley, for your comprehensive talk. There is time for one question or two short questions.
Mark.
Bill, how does leptin modify some of these pathways?
Excellent question. The leptin receptors are on the kisspeptin neurons, not on the GnRH neurons. So they shut off the upstream pathway, and mutations in it. It's always been known that the second messenger pathways can shut off nutrition versus reproduction. There was a differential specific mutation in that. Those mediate through the kisspeptin. It's an excellent example of nobody could figure out where leptin and GnRH work because the receptors weren't there that were necessary. And only when you understand the full ensemble of genes, you figure that's how it works. And I think this is the tip of a bigger iceberg. We're going to have that's why I showed the fields of study. These are going to be indirect, autocrine, paracrine, complicated relationships. And sometimes designing therapies without that knowledge is going to, and leptin hasn't worked in certain areas, when they've tried it anyway. Ron?
Beautiful talk. And from a practical point of view, can you activate that FGF it looks like it's a heterodimeric receptor.
Yes.
And therefore it combined multiple FGF genes. You implied those are also being bound as combinations of FGFs.
That's a great question. It's not like the orphan receptors. They seem to be homozygous dimers, as best we can tell.
Homozygous, okay.
Yes, and it's the clotho and the other ones that add directionality to it. The story about the receptors becoming much more like the transcription factors. There's more and more guidance molecules that are outside of the heterodimer. Now the GPCRs, there is some evidence of heterodimerization. And in fact, that comes out where some receptors can rescue each other. We have studies about heterozygosity where you can have a mutation in the ligand binding, a mutation in the effector site. You put them together and you don't have any mutation because each compensates for the other. But for FGF, they seem to be homodimers.
And can injected FGFs influence that axis?
Well, it's in the brain, so you'd have to deliver it locally to this. Most of them are skeletal, so there is a possibility, I suppose, of doing this, but I don't know any data about that.
A fair number of FGFs, I think, do get to the brain now.
Yes, no question about it. The ligands, yes.
Bill, you implied that some of the GnRH neuron defects, if you want to look at it that way, or gene mutations, allowed the GnRH neurons to get to the hypothalamus, and then what happens? Why aren't they working after that? Is it because they don't connect, or is it because they don't survive? What's going on?
Great question. If you look at, from the clinical point of view, a gain of function, a good example would be an FGF receptor. There are some activating FGF receptors, and they cause synostoses. So they have cruzons and a variety of these disorders. But from our point of view, if the GnRH neurons matured too early, they would not arise. And therefore, they would look like loss of functions to us in our system, but while they'd be gain of functions biochemically. So I think a lot of them are that when they've looked at these systems, FGF being one, the number of surviving GnRH neurons is small. The number that migrate is small, and there's a certain attrition along the way. And that's probably how it has a footprint in both pathways. The number of GnRH receptors that are born are small, and then they migrate, and there's even fewer of them. It's a good question.
Thank you very much, Dr. Crowley.